Good morning, all. Welcome back to our SRN BGNR Government Degree College Khammam official YouTube channel. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. Today, I am going to explain about some fundamentals of enzymes in enzymology. In fact, this is the part 2 video of this topic. In the previous video lesson, I have covered some topics in enzymology. As I said in the previous video lesson, this enzymology topic is present in syllabus of second year biochemistry subject, third semester, paper 3. Okay, and in the previous video lesson, I explained some of the topics, subtopics in this enzymology, like introduction to biocatalysis, differences between chemical and biological catalysis, and then the nomenclature process and classification of enzymes, and we have covered enzyme specificity also. These three subtopics have been covered in the part 1 video lesson of enzymology. Before going into details of remaining subtopics of enzymology, I would like to once again summarize the subtopics we have covered so far in the previous video lesson. First of all, the definition of enzyme I explained. As we all know that enzymes are biocatalysts. Before going to see the definition of enzymes, we have to know what is a catalyst. A catalyst is a substance that increases rate of a chemical reaction without itself undergoing any permanent chemical change. So that means the structure of a catalyst will be same before the chemical reaction and after the chemical reaction also, but it will increase the rate of the reaction. So that is the role of a catalyst. Without catalyst, the reaction may be very slow and the products may be very less, the product yield may be very less, and it may require very some conditions, extreme conditions like temperature, pressure and pH and like that. So catalysts are really helpful in carrying out chemical reactions at mild conditions and with higher product yield. In the same way, enzymes are also very important in biological systems where thousands, even lakhs of chemical reactions takes place simultaneously within a given environment. Say for example, in a cell, every cell continuously carry out thousands of different chemical reactions per second simultaneously. So each enzyme, each chemical reaction is catalyzed by a particular enzyme, so that no two chemical reactions can interfere with each other. Every single reaction proceeds on its own way, and there is no interference between them because of the specificity of the enzymes. Suppose if you don't have any enzymes in the biological system, then what happens? We have an example already explained. I explained in the previous class. In this example, if we take the oxidation of sugar, it requires a few seconds to complete the reaction. But without an enzyme, it takes even years, and the product may be very small, and the liberated energy may not be captured easily by us for our utilization. So that is the importance of enzymes. Without enzymes, we cannot imagine the reactions plenty of reaction that takes place in each and every cell of organisms. So that is why it is very important to study about the enzymes. And next we have seen of course the definition of enzymes and the differences between chemical catalyst and biological catalyst, that is enzyme. Chemical catalyst means they are inorganic in nature. They usually used in organic or inorganic laboratories outside the cells, outside the living organisms. When compared those chemical catalysts with the biological catalysts that are enzymes, we will have some differences like the chemical catalysts are simple inorganic molecules, but enzymes are complex proteins. Enzymes are made up of, actually they are proteins, they are made up of with sequence of amino acids, and the molecular weight of catalyst is very low. Most of the times these catalysts are metal ions, but whereas in case of enzymes, because they are polymers of amino acids, the molecular weight of enzymes is very high when compared to the chemical catalyst, and the reaction rates are, in fact they are very fast when compared with the non-catalytic reactions. The chemical catalytic reactions are very fast, but when compare the reactions carried in the presence of chemical catalyst is slower than the enzyme catalyzed reactions. So the enzyme catalyzed reactions are very fast, even 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 more faster than the chemical catalytic reactions. And specificity, the chemical catalysts are not specific. They carry out some one particular of reaction, but the reactant specificity may not be there. Different reactants may be used with one particular chemical catalyst to carry out a specific reaction. But in case of enzymes, enzymes are very specific to their substrates and their reaction also. So they are highly specific. Because of the specificity only, the chemical reactions within a cell cannot interfere with each other. Everything goes on its way. And the conditions for chemical catalysts, the conditions may require like high temperatures and pressure, but in enzymatic reactions we need only mild conditions, because all those chemical reaction takes place inside the living systems. Inside the living system we cannot expect higher temperatures like 100 degrees centigrade or 200 degrees centigrade, and high pressures or high pH or low pH. All those conditions are not possible, because that affects the life of organisms. So we will have a very narrow range of physiological conditions, like the temperature. If we say the temperature of an organism, that is suppose human being, if you take example human being as an example, human being has optimum temperature around 37 degrees centigrade. Above or below this 37 degrees centigrade physiological temperature, what happens? That will drastically affect the health, seriously affect the health of the individual. So therefore, whatever reaction takes place inside that human being should happen at that temperature only. So that is why enzymes are capable like that. They can carry out any reaction, their own reaction, within the range of physiological conditions. So that is the importance of enzymes. Okay. And next we already learned about the nomenclature process of enzymes. International Union of Biochemistry has appointed an enzyme commission, EC commission, in 1961 for the classification and nomenclature of enzymes. In the classification, they have suggested 6 different classes for all the enzymes based on their reactions that they carry. Okay. So some of the enzymes which transfer hydrogens or electrons, they are no comes under the class of oxidoreductases. They do both oxidation and reductions reactions. So they are called oxidation and reduction reactions, and the class is oxidoreductases. Lactate dehydrogenase, alcohol dehydrogenase, all these enzymes comes under the class of oxidoreductases. And transferases, second class is transferases, means they transfer functional groups from one substance, one molecule to another molecule. They are called transferases. Some like group transfers. If we say that phosphate group transfer from ATP to glucose, so then exokinase is an transferase enzyme. And the third class is hydrolases, which hydrolyze polymers like carbohydrates, lipids, proteins, and nucleic acids. And fourth class is lyases, which carry out reactions which involve in reactions where addition or removal of small molecules, small groups, small molecules like water, ammonia, carbon dioxide, etc. takes place. And fifth group is isomerases, which are involved in the isomerization reactions. And last group is ligases. These ligases are also known as synthetic enzymes, which ligate or join two small molecules to form a large molecule. So these six classes are actually classified, suggested by EC. So based on this classification, the nomenclature also set up by the EC Commission. According to the EC Commission, they have given one particular numbering system for each and every enzyme. So each and every enzyme will have one EC number. In EC number, we will have four different parts separated by full stops. The first number, it represents the class that the enzyme comes under. Suppose the one example we have taken here, lactate dehydrogenase. It catalyzes the reaction of formation of lactate from pyruvate. It depends on NAD plus for electron transfer. Pyruvate receives two electrons from NADH and it becomes lactate. And in the same way, the lactate becomes pyruvate by giving the two electrons to NAD plus to become the NAD plus becomes NADH. So both sides, this is reversible reaction that is carried out by lactate dehydrogenase. So that is why it comes under oxidoreductases class. And the second number is for subclass of the main class. And third number, that is subclass. In that these subclass are different for different electron acceptors. And fourth and that is the last number, that is for the number is given for one particular enzyme. Each and enzyme will have a specific number. That specific number is the fourth digit. So this is about the classification and nomenclature process. And the last topic we have covered so far is specificity of the enzyme. We have three levels of specificity in enzymes. Some of the enzymes are stereospecific. They recognize their own substrates, and along with the substrates, they also see whether they are a particular stereotype or not. Suppose if alpha amino acid oxidase is our enzyme that recognizes only, sorry, L amino acid oxidase recognizes only L amino acid but not the D amino acid. Okay, so the substrate binding site, which is known as active site, will be formed in such a way that L amino acid only fits in that active site, not the D amino acid. Like the right hand glove, if we take and if you try to put our left hand in the right hand glove, it does not fit. But both hands look same. But in fact, they are uh, mirror image of one another. So in the same way, some of the enzymes are very specific. Uh, the specificity is more, even they differentiate between different stereotypes also. And second specificity is reaction specificity. So one single substrate may undergo different types of reactions which are catalyzed by different enzymes. Suppose one amino acid is there, it may undergo a transamination or deamination, a decarboxylation reaction or a racemization reaction. Each and every reaction requires a particular enzyme. So that is why these enzymes are reaction specific. All the enzymes are acting on same amino acid, but each enzyme is catalyzing different reactions. So they are reaction specific. And third specificity is substrate specificity. So this is very important specificity when we discuss about enzymes, because enzymes are very specific for their substrates. This substrate specificity again classified, subdivided into three categories. First one is absolute substrate specificity, that means 100% substrate specificity. One particular enzyme recognizes only that substrate, not the other substrates or any other similar substrates. So here the example is glucokinase. Glucokinase strictly converts glucose to glucose 6 phosphate. It does not bind any, it does not catalyze or bind the any other substrate which are similar in structure with the glucose. Relative substrates means these enzymes bind different substrates because they will see, they look into the substrates for specific group or a specific bond. If that specific group or specific bond is there, just it recognizes that substrate. So in that way, more than one substrate may have same functional group or same bond. So in that way, these enzymes recognizes more than one substrates based on a specific group or a bond present in it. And third specificity is broad specificity, where similar kind of substrates also recognized by a single enzyme. The example for this is hexokinase. That hexokinase uh, acts on both acts on glucose, fructose, mannose, and galactosamine. All these substrates are acted upon by the hexokinase. So hexokinase specificity is very broad specificity. The next topic we are going to see is active site of enzymes. Earlier in this video, I explained that active site is the site where the substrate binds. The definition of active site is the active site of the enzyme represents a small region as the small region at which the substrate binds and participates in the catalysis process. The substrate binds to the enzyme. We have one diagram in this slide. The black one is the enzyme which is protein in nature. The size of the enzyme is very high. It is very large in size, and we have the region, some crew region is there to which the substrate bind. So we have the substrate on the top of this diagram with green color. Okay, in this diagram, this is the enzyme. Enzyme is made up of with sequence of amino acids. Suppose this is a sequence of amino acids. We have one amino acid here, 2, 3, 4, 5, 6, 7, like that. Sometimes the protein or the enzyme may have some hundreds, several hundreds of amino acids that are aligned in a single string like structure. So this a length of amino acid sequence folds upon itself and forms like a ball, and it becomes an enzyme, a protein. Okay, suppose it is folded upon itself and formed a ball like structure. This ball like structure is now called enzyme if it binds substrates and carry out a catalytic reaction. So this is an enzyme. This thing is an enzyme. Okay, so in enzyme what happens, different amino acids, suppose the amino acid, the first amino acid may, the first amino acid here may interact with another amino acid here. Okay, so in this way the amino acids at different places in the same region, same sequence may interact with each other by non-covalent interactions. Non-covalent interactions. Okay, because of those non-covalent interactions, the interaction between amino acids are not rigid. They can be easily broken down and they can easily reformed. Okay, so that is why this enzyme is a dynamic molecule. It can change its shape, it can change its conformation. Okay, so now we have active site. So this region is the active site. Okay, so this region is our active site. Now the substrate, this one is our substrate. This substrate binds to the active site of the enzyme. Once it binds the active site, we will have a complex. This is the complex, that is enzyme substrate complex. Now what happens, this enzyme should carry out a catalytic reaction by which the substrate becomes a product. Okay, before that what happens, this enzyme slowly, this enzyme will convert the substrate into a transition state. It is not actually first of all it convert into a transition state which is intermediate to substrate and product, which it the structure of the transition state is between substrate and product. So then it converts the transition state into product. So these are two products. From one substrate we are getting two products. Okay, so now what happens, once the product is formed, the product, the structure of the products are different from the substrate. So that is why these products can no longer bind to the active site of the enzyme. They easily liberated from the active site. So now we have this substrate, free enzyme and products. Now this free enzyme comes back and again binds another substrate, and that forms a substrate enzyme complex, and the substrate becomes a product. So this cycle takes place for several hundreds, even thousands of times, and each one enzyme molecule convert thousands or sometimes lakhs of substrate molecules to products. So in a typical reaction, the active site binds, the active site of the enzyme binds substrate, and it gives a enzyme substrate complex, and enzyme substrate complex, after catalytic mechanism, the substrate is converted to product, and enzyme products or will be separated with each other. The enzyme once again comes back and binds to another substrate and complete another cycle of enzymatic reaction. So that is called active site. When we look at the features of active site, here I already mentioned that active site, the enzyme, in fact, enzyme is made up of with amino acids. Okay, sequence of amino acids. They are the sequence of amino acids, and these amino acids interact with each other to form a three-dimensional folded structure. Okay, three-dimensional folded structure to form a functional enzyme. The second point is enzymes are specific in their function. They specifically bind specific substrate, and only they catalyze their own specific reaction but not other reactions. If we take example of glucokinase, that strictly binds glucose and it converts to glucose 6 phosphate only. It doesn't bind, it cannot bind other sugars like fructose, galactose, or anything else. If it binds glucose, then it converts only that glucose into glucose 6 phosphate, not into other products like fructose or glucose 1 phosphate or anything else. So that means the enzymes, most of the enzymes are substrate specific and reaction specific. That this specificity is the feature of active site. Okay, and the next important point is the association, the interaction between substrate and the active site of the enzyme is by weak non-covalent interaction. There is no permanent covalent interaction between active site and substrate. So that is why the substrate easily binds to the active site, and once it is converted to product, it easily dissociates. So that is why the reaction is very quick. Suppose if substrate binds to active site with a covalent interaction, what happens? Once the product is formed, that covalent interaction must be broken down in order to release the product from the enzyme. That requires a lot of energy, and it requires time also, time consuming process. So that is why the interaction between substrate and its active site is by non-covalent interactions only, very weak interaction. So that is why easily binds, easily dissociates, making the enzyme very fast. And next point is the active site is not rigid in structure and shape. Okay, this is very important point because the active site is not rigid. So first of all, it will be in such a way that not rigid means that active site is flexible. The shape of the active site may change by the time the substrate interaction. The shape of active site is not a fixed shape, not a rigid shape. So that is why when specific substrate comes in contact with the active site, the shape of the active site will be changed in such a way that substrate fits into the active site. And last point is the active site of enzyme actually contains two sites. One is substrate binding site where the substrate binds by non-covalent interactions, and another one is catalytic site. Catalytic site is the site which carry out the catalytic reaction. In the previous slide, if you look at here, so this is active site. Active site is binding the substrate, but actually this one here, this region is the catalytic region, catalytic site, which breaks the bond between these two parts of the substrate. Okay, so because of this site only, the substrate is broken down into two parts which are products. Okay, so this is what the catalytic site, substrate binding site for the binding of substrate to active site, and catalytic site for the reaction. Okay, this is about the active site of the enzyme. And next we move on to another subtopic, that is activation energy and transition state. When we start learning about enzymes, the first question that we, we may come across in our mind is how these enzymes catalyze the reactions which requires very extreme conditions in their absence, in the absence of enzymes. Suppose if we don't use enzymes, the same reaction requires extreme condition. If you use the enzymes, the same reaction will be carried out at normal conditions. So what is the reason behind this efficiency of enzymes? To answer this question, the secret behind the efficiency of enzyme is we have to learn about two important things of any reaction. First one is activation energy and transition state. Second one is transition state. Suppose when the reactants are heated, they will get activation energy. The enzymes are the molecules which reduces the activation energy and causes the reaction to proceed at a lower temperature. So what does it mean? That means for every reaction to take place, the reactants must be converted into transition state. For that, it requires some amount of energy, external energy. So that energy, that amount of energy which is required by the reactants to reach the transition state is known as activation energy. So we have to activate the reactants, then only that reactants becomes products. In case of enzymes, the substrate must be activated so that it reaches a state of transition. From the transition state, it becomes product. So the amount of energy that should be externally given to the substrate to become a transition state is known as activation energy. If you don't use enzymes in reaction, what happens? It requires a lot of activation energy. But when we use enzymes, what happens? The enzyme will lower the need of activation energy. Suppose in a reaction, non-enzymatic reaction, the substrate need 100 units of activation energy. Suppose when we use enzymes, that 100 units of activation energy may come down to 10 units or 5 units. So in that way, we are reducing, the enzymes are reducing the need of activation energy. Once the activation energy is reduced, the substrate is easily converted to transition state product, and then the transition state becomes a product. So in that way, enzyme will reduce the activation energy to proceed the reaction from substrate to product. So that is why the enzymatic reaction is very fast. It requires less energy, and because of the active site, it is very specific. And another important term is transition state. What is transition state? A transition state means it is an intermediate state between substrate and product. Substrate never be converted into a product directly. Substrate first converted into a transition state, and then it is converted to a product. The reactants and products have specific energies. In order to transform the reactants into products, the reactants would have to go through a transition state which usually higher in energy. What does it means? Suppose if you look at this graph, what we will observe, on the y-axis we have energy and x-axis the progress of the reaction. So the reactants, the energy of reactants is around here. The energy of reactants is around here, and the energies of products is around here. So the energy of reactants is higher than the product. So in this case, the reactants must be easily converted to product, because the movement is downhill movement. Higher energy reactants are being converted into lower energy products. So it should take place spontaneously. But in fact, it does not take place spontaneously, because the reactants first of all must be converted, or uh, must reach a transition state, the intermediate state. The transition state is here. Okay. In a enzymatic reaction or uncatalyzed reaction, the transition state, the energy required for the transition state is more. So that is why we have to provide a lot of energy to the reactants in order to reach the transition state. So that is why we may require higher temperatures, higher pressures, or some extreme conditions. What happens when we use the enzymes? The enzymes will lower the activation energy. So that means now what happens? The energy required to reach the transition state in an enzyme catalyzed reaction is very less. So at normal conditions and a normal physiological temperature, pressure, all those things, it easily takes place. So because of the reduction means here non-catalyzed, this is catalyzed. The difference between these two energies is known as the reduction in the activation energy. So this is the activation energy to reach the reactants to transition state. The activation energy is lowered in an enzymatic reaction. So this is the difference from here to here. Okay, in the presence of enzyme, the activation energy is lowered, so that the reactants easily reach the transition state, and from that they will be converted to product. So the enzymes are the molecules which reduce the activation energy in order to proceed the reaction faster. So this is how the enzymes requires very mild conditions to carry out a reaction which requires extreme conditions in the absence of enzyme or any catalyst. So in this chapter, in this video lesson, we have covered about the active site, its nature, and how the active site is formed, and how the active site plays an important role in the specificity of enzyme, and what are the parts present in the active site. We have substrate binding site, and another one is catalytic site, and of course the very important subtopic in this enzymology is the activation energy and transition state. By lowering the activation energy, the enzymes perform the catalytic reactions with speed. Okay, so that is the main mechanism which influences the efficiency of the enzyme. Yeah. So in the next class, last class of this topic, that is part 3, we are going to discuss about the last subtopic, which is the interaction between enzyme and substrate. We have different models explained by, proposed by different scientists. Okay, we will cover it in the next lesson, and thank you all.